Thank you all. Great to start off this series of presentations today. I hope we don't have just a super bloom here today, but just also a super presentation day. So like you said, I'm Dr. Moyers. Just started here last month at the Angeles Clinic. I'm excited to lead off this group. So as an outline of what we'll talk about today briefly in my my discussion, we'll first start out the rationale for some combinations and what's historically been seen already with the single agents. Next, we'll move on to some of the improved combinations and discuss nivo and ipi, as well as nivo and relatlimab. And then also at the end, discuss a couple of the up and coming things and things that are going on at our clinic here. So melanoma tumors are comprised not only of cancer cells, but of a host of immune cells in the so-called tumor microenvironment. The balance of this tumor microenvironment is dependent upon a host of factors. Some of the cells cause an immune suppressive environment, such as the regulatory T cells, pro-tumor T cells, and myeloid-derived suppressor cells, while cytotoxic T cells and T helper cells are positive immune regulators. The goal of immunotherapy is to tip the scales in the direction of the tumor death for a positive immune regulator environment to elicit tumor death. Two of the first checkpoints discovered were found to block anti-CTLA-4 and anti-PD-1. This is a schematic representation of the immune cell types primarily affected by anti-CTLA-4 in the red arrows and anti-PD-1 in the blue arrows, as reported in humans. Notably, most of these cell types express the targets of immune checkpoint blockade, potentially explaining their preferential modulation after immune checkpoint blockade. Anti-CTLA-4 blockade induces T helper 1 cells to effector and T while counteracting T regulatory function and possibly expanding X cells. So this led to the trial of first checkpoint inhibitor that was approved, the anti-CTLA-4 ipilimumab. It was a dramatic improvement in the treatment of metastatic melanoma compared to cytotoxic chemotherapy and led to some durable survival and appeared to have some that had to have some dose dependency. However, only about a fourth to a fifth of patients had a long-term survival effect, as we see in that tail at the right end of the graph. Next came anti-PD-1 with nivolumab and pembrolizumab in several key trials. And one of the ones I'll show real quick in Keynote 006 in the long-term follow-up, anti-CTLA-4 ipilimumab was compared to pembrolizumab at 2 different dosing regimens. PD-1 pembrolizumab combined cohorts were found to improve median overall survival of 32.7 months versus 15.9 months compared to anti-CTLA-4 in the long-term follow-up. This led rationally to think, well, if 1 is good, maybe 2 better. So the combination of anti-CTLA-4 and anti-PD-1 was also compared to single agents. Patients were assigned to 3 treatment arms, equal ratios of the 3 treatments to nivolumab alone, ipilimumab alone, or the combined nivo and initial results showed superiority of the combination over either drug in single agent form, with the highest objective response rate illustrating tumor shrinkage in over half of patients. 50% of patients to just over 15% for, but by over 15% difference to nivolumab. However, as with any effective treatment, side effects occurred and occurred universally, in all 3 groups to some degree. Serious side effects graded as 3 or 4 occurred in most of the combination patients at just under 70% and nivolumab at 40%. Side effects led to discontinuation in about 30% of the combination group and only 5% of the single agent nivo group. Since this trial started a while back, we now have long-term survival follow-up at 6.5 years. Median overall survival is just over 6 years for the combination, with a landmark survival at 6.5 years of just under 50% at 49%. This combination was also in patients with brain metastasis, in a cohort of 101 patients with asymptomatic metastasis, and a cohort of 18 patients with asymptomatic metastasis in the CheckMate 204 study. In the asymptomatic cohort, the intracranial benefit rate, or the shrinkage of brain metastasis, was 57%, and 30% of the cohort achieving a complete response of the brain metastasis. The landmark survival at 36 months was 71.9% in the asymptomatic cohort, and only 18.9% in the symptomatic cohort. So we see that this is not only effective in the body, but in the brain as well. So these studies show that the combination dual checkpoint inhibitor was superior to single agents. However, as we discussed, the treatment's quite toxic. It leads to lots of side effects. And so is there a path to getting a better side effect profile for patients? So CheckMate 511, tested a flip dose of ipilimumab and nivolumab using 1 milligram per kilogram of ipilimumab and 3 milligrams per kilogram of nivolumab instead of the inverse. The initial dose of for the initial dose of the 4 immunotherapies. As seen on the right, the median progression-free survival and overall survival were not significantly different at this first initial presentation, with median PFS around 9 months and median overall survival not reached for either group. But here's the kicker, is that the side effect profile was substantially better for the quote-unquote flip dose, the Nivo 3 IPI-1, at 34% serious or grade 3 to 5 events, whereas only is increased number of 48%, so nearly half had serious side effects on the IPI-3 arm. Next, we moved on to a novel checkpoint. LAG-3 regulates a checkpoint pathway that limits the activity of T cells, as well as may lead to resistance in immunotherapy experienced patients. The relatlimab anti-LAG-3 was an anti-LAG-3 agent that was tested in combination with nivolumab. And we saw this in Relativity 047. It had an every 4-week dose regimen comparing nivolumab alone with nivolumab and relatlimab. Progression-free survival was different and met statistical significance over nivolumab alone, when overall survival was longer in the relatlimab as well, but not significant yet. And then we looked at, does the LAG-3, how much LAG-3 is on the cells affect how well it works? And that if the LAG-3 expression was greater than 1%, there was a improved survival in the combination. However, only a trend towards improvement was seen in the combination arm. We also saw that the side effect profile showed that the Nivo and relatlimab arm was not nearly as difference of toxicity as compared to nivolumab alone as we saw with the Nivo and IPI. And so serious side effects occurred in about 20% of the combination arm, only 11% of the single agent arm. And so putting it all together, I like this slide I borrowed from Dr. Dowd that shows the x-axis on the bottom, the progression-free survival, and on the y-axis, the grade 3 or 4 serious events. And so we saw that while CheckMate 6-7, the Nivo and IPI at the standard dose was the most effective and most toxic, when you either switch it to the flip dose or add relatlimab instead of ipilimumab to nivolumab, we see nearly equivalent progression-free survival, but significantly reduced side effects. So is there something on the horizon about to make a big splash? Do we need some more immunotherapy combinations? While immunotherapy has allowed the field of oncology to turn a critical corner, where there's even long-term survival and even durable cures, many patients still don't benefit from these checkpoint inhibitor therapies. The thought is immune suppressive environment resists reversal through several mechanisms. One can suppress suppressive cytokines or anti-inflammatory immune molecules. 2, lack of antigen presentation causing the immune system to not properly recognize cancer as foreign, programmed cell death mechanisms, or a hostile metabolic state. Therefore, new therapies are needed to overcome these obstacles. Our own Dr. Hamid presented in Paris the phase 1 results of the novel combination of nivolumab as well as fianlimab, an anti-LAG3 checkpoint inhibitor that enrolled patients here at the Angeles Clinic. In this study, 76% of patients had a level of tumor reduction with the best response with median progression-free survival of 24.0 months for the PD-1 naive patients. We're awaiting release on more data of this exciting combination. Last month, Dr. Doomer presented at the AACR conference, Keymaker U02. This trial compared 3 different neoadjuvant combinations, two combinations and one control arm of pembrolizumab or pembrolizumab plus intratumoral jabexarev or pembrolizumab and vibostolimab, another checkpoint and anti-TIGIT antibody. The pembro plus vivo arm had responses in 81% of patients in comparison to the pembro arm had about 73%. Obviously, these numbers are way too small given the number of patients in each group to make comparisons between the arms. However, the pembro plus vivo is pretty encouraging and exciting. Another combination that we're excited here at the Angeles Clinic that's enrolling uses sirolimus. This is an IL-6 receptor blocking antibody. This trial is used in combination with nivo and ipi in unresectable and metastatic melanomas. So what's next? Thus far, we've discussed the effects of checkpoint inhibitors as being anti-tumor effect of cancer. How are the other points in the cancer immunity cycle where immunotherapy may be useful? Oncolytic viruses and peptides can cause tumor death and release tumor-driven antigens. And there's also a potential to affect trafficking and infiltration of activated T cells into the tumors. Other places that might be helpful. Others will be talking about further strategies to treat melanoma today. And some of them play directly to target the cells through tumor death with cytotoxic systemic chemotherapy, radiation therapy, molecular targeted agents, epigenetic modifiers, all directly affect the tumor. Indirect methods to modify the tumor microenvironment and the tumor to favor anti-tumor immunity include enhancing the effect of effector T cells and antigen-presenting cells, or trying to block the inhibiting agents such as regulatory T cells. Another hot topic is the modification of the gut microbiome, the local vasculature of the immune system, the cytokine milieu, altering cellular metabolism. It's likely that this complex interplay is not acting in isolation, and all these arms are dependent on one another. We started out with the treatments aimed at PD-1 and CTLA-4, but as we can see, there's lots more potential to target in the cancer environment for melanoma. Thank you for your time. I'm glad to give the microphone over to Dr. Memi.